Okay, well, thanks very much for the introduction and the opportunity to be here today. It's a real honor and nice to see all my colleagues and friends as well. It's also a pleasure too, because Neil was really channeling me when we were talking about his introduction today. I also have a historical view on it as well. But for what I'm going to talk about, I think it's really important to look back at some of those early pharmacogenetic discoveries, because that really helps inform ethnic discovery in pharmacogenetics, and talk a little bit about population differences in drug response variability, and then spend most of the time on multi-ethnic pharmacogenomic allele discovery, as this is something that I've been kind of interested in for the last few years, and I know some of you are as well too, particularly in the fields of cardiovascular pharmacogenomics. Do a little divergent technical example with a CYP2D6 long read sequencing, and then go into one of our favorite populations in New York City, which is the Ashkenazi Jewish population, and kind of end off with how this is important, not just for discovery, but I think also dovetailing what we talked about before lunch, which was clinical implementation and how important this topic really is for that. So after the introduction to the session, I don't think we need to go too much detail through this, but for anybody interested in the historical views of pharmacogenomics, Urs Meyer's review from over 10 years ago is still excellent read on the topic. But in addition to Sir Archibald Garrod, I would also like to suggest, on top of what's already been discussed, is that some of these major discoveries from a long time ago were really helped or enabled by the racial differences in their occurrence. One of the first being the taste bitterness with phenylthiocarbamide that was observed with racial differences in its incidence. And of course, the G6PD discovery among African American soldiers and how increased the frequency that was over the Caucasian soldiers. And then later on, the isoniacid TB induced acetylator phenotypes, the differences between populations there later found out to be due to NAT2 deficient alleles. So just really underscoring that ethnic differences in these phenotypes can really help enable discovery for pharmacogenomic alleles. So another individual I think warrants some discussion is Werner Callow from the University of Toronto, who was one of the first, I think, to really talk about drug metabolism differences between populations and those ethnic differences between them. These are two really great reviews from the 80s and 90s that I reviewed in the last couple of weeks. And what he really underscored was that these drug effects, they can vary among individuals, but they can also vary between populations. And that these differences are measurable and observable at different levels, at pharmacokinetic measurements, pharmacodynamic measurements, and that all of these differences can really help influence efficacy, safety, as well as dosing issues. But when we look at the distribution curves of these measurements, you can really see that the inter-individual variability is much wider than that between the inter-ethnic variability, as we'll see in the next slide. So the mechanism behind these ethnic differences in population drug response variability can be a number of things, as I'm sure you all know. Some of them being environmental and social factors, diet, the various practices of medicine in these populations, patterns of concomitant medication use, and of course what we're, I think, more so interested in is the complex trait or Mendelian genetics underlying these differences, which ultimately I think is really driven by the differences in allele frequencies between these populations. So some of the enzymes that he really focused on and characterized back in the 70s and 80s were the esterases, the peroxygenases, dehydrogenases, acetylation, and others like that. I was going to show some of the raw data from these early studies in the 70s, but the terms that they used to describe populations in the 70s were so politically incorrect that I thought I was going to get beat up if I said those words today. So just to suffice to say that he was really, I think, forward-thinking, though, too, and that he said over 30 years ago that you could actually measure the differences between the population drug response variability if you overlay it with a genetic map or the genetic distances between some of these populations. I will show one raw data slide from the 70s, though, looking at aspirin esterase activity between two different populations, the Ghanaians on the top and the European British on the bottom. And as you can see, once again, we have that large interpatient variability and only slight differences in the median between the different ethnicities. But what he underscored was that despite the fact that the majority of individuals are all going to have a therapeutic benefit from these overlapping enzyme activities, that's not what we should be focusing on. What we should be focusing on are the 10 individuals from the Ghanaian population who likely aren't going to have any efficacy from this medication. And then the 5 individuals in the British UK site who are at much more higher risk for toxicity. Again, focusing on these extreme phenotype outliers, which I'd like to just overlap with 35 years later. One of the most kind of interesting and exciting discovery tools that we have right now is these exome sequencing, genome sequencing strategies among extreme phenotype populations. So I think the last slide on kind of the history would be, again, underscoring the HLA alleles and then how they have really informed us on the magnitude differences and or the phenotype differences between the same alleles in different populations. And we can see that by the odds ratios of allopurinol-induced Stevens-Johnson syndrome with the star 5801 allele. Now you can see a really remarkable signal, high odds ratios in the East Asians, but it really gets diminished in the European populations. In contrast, we have a difference not just in the magnitude, but in the actual phenotype with the star 3101 allele and carbamazepine, where in the Europeans, we have a strong association with the hypersensitivity syndrome plus the severe Stevens-Johnson's, whereas in the other populations, we have just the association with the hypersensitivity syndrome. And finally, with the 1502 allele in phenytoin-induced Stevens-Johnson syndrome, this is kind of a classic example of where the minor allele frequency is really restricted to the East Asians and the Middle Eastern populations and is absent in Africans and Europeans. Fast forwarding to today, I think one of the nicest examples we've had in the last year has been Jun Yang and others at St. Jude's, their beautiful work on NUD-T15 and mercaptopurine toxicity in childhood ALL, taking advantage or observing that there's differences in the mercaptopurine dose intensity across ethnicities. And after a genome-wide association study, successfully identifying a second signal in addition to TPMT, which was a much higher allele frequency in East Asians and Hispanics compared to Africans and Europeans, and strongly correlated with dose intensity in these childhood ALL cases. And I believe this is on the fast track for a CPIC guideline in the upcoming future. So as I mentioned, part of our focus is on cardiovascular pharmacogenomics. One of the examples that we have spent time on is clopidogrel. I'm sure I don't need to explain the variability in response to this audience, but most of the genes involved in the clopidogrel pathway through the absorption, the esterification, the biotransformation in the liver, through the P450 system, as well as the target where the active metabolite binds irreversibly to the P2Y12 receptor, as well as the integrins. These have all been studied for variability or genetic variation that can inform drug response, but the most robust and reproducible signal has been, of course, with CYP2C19 and the no functional alleles within that gene. So we've studied the frequencies of these alleles across the 2C19 region fairly extensively in 5 populations from the New York City region. We have Africans, African Americans, Hispanic, Asians, Europeans, and different Jewish subpopulations. This is taken from the CPIC supplement, though, however. And I think what most people know is that star 2 is the allele that really everybody focuses on. Star 3 is found a little bit more common in East Asians. But then, of course, there's star 17, which is quite common in certain populations like Africans, Europeans, and in East Asians as well. But there's a battery of rare variants across the gene that have biologically been shown to have a loss of function, but they're just at a very, very low frequency. So we surveyed all of these alleles plus about another 20 more star alleles across all of our New York populations and made an observation a couple of years ago that we thought was fairly important when we considered the combination of common and rare variants. And that was, we found the star 17 allele always to be tracking with star 4 when we found star 4. So star 4 is a loss of function allele that abolishes the translation start site of the gene in the first exon. And luckily, these two variants are very close together. They're less than a kb apart, so that allowed us to develop an allele-specific PCR assay to infer the phase of these two variants when they were detected. We submitted this to the P450 nomenclature site, and this subsequently was called star 4b. And so that throughout all the different populations that we surveyed, it was the most common, or actually almost predominantly located in the Ashkenazi Jewish population. with an allele frequency of about 20 or 2%. And although that's not the most common allele compared to STAR2 and things like that, we think it's actually very important because most of the FDA-approved 2C19 genotyping assays only include STAR2, STAR3, and STAR17. So some of those STAR17 individuals are actually not rapid or ultra-rapid metabolizers. They're most likely going to be intermediate or poor metabolizers based on the STAR4B allele. So these people would be missed by genotyping with an FDA-approved assay. So in addition to 2C19, we've also explored more discovery for clopidogrel response variability. And for that, we looked at the trait of ex vivo platelet aggregation using both PRUs and residual platelet activity. And this is with a collaboration with my friend and colleague, Jean-Sébastien Hulot from Paris, who also has an appointment at Mount Sinai. And for this cohort, we used a multi-center study called ONASYST that previously has been published, hospitals throughout France. We looked at the sensitive and resistant outliers based on their definitions shown here with low on-treatment platelet reactivity and high on-treatment platelet reactivity. We made sure to screen them for 2C19 first, so they were all wild type. And then subjected the two outliers to exome sequencing, ranked the variants as kind of thoughtfully as we could. That was a bit of a lucky happenstance, as it were. But and then take the top ranking variants and genotype them through a multiplex panel across the entire cohort. And what we found, I think we got kind of lucky. We did find B4GALT2, two variants in the gene that was strongly associated with low on-treatment platelet reactivity, it was a marker of sensitivity, clopidogrel sensitivity, which is opposite to CYP2C19, and that carriers had a much lower on-treatment platelet reactivity. So when we regressed this in a linear regression with other variables known to influence platelet aggregation, such as 2C19, diabetes, and age, it still remained significant, which was encouraging. And then in our replication cohort, which was also European ancestry. So I should underscore that the French cohort was 99% Europeans, Caucasians. Our replication cohort was from New York City, which was also 100% European in this sense. And so we got a nice replication of the signal of B4GALT2, even when in the presence of CYP2C19 and other clinical variables. Now I highlight the ethnicity here for the following slide. So our hypothesis is that B4GALT2 actually influences platelet aggregation and adhesion because it's a galactosyl transferase. So it modifies proteins as they're excreted to the platelet surface, which is most likely, or what we hope or think, is that this is modifying the actual integrins that are involved in platelet adhesion. However, the allele frequencies are fairly high frequencies, around anywhere from 6 to 12% through the exact database on any given population, as shown here. But when we actually did the replication in the New York City cohort, we had a much larger cohort available to us. We restricted it to the Europeans in the paper. But when we included other ethnicities, that the Africans, we have a lot of South Asians in this cohort, as well as a high frequency of Latinos, the signal was completely attenuated. Suggesting that this is not the functional variant. This is actually a synonymous variant in B4GALT2. So it's likely that this variant is not going to be of utility in some of these other populations. And the reason for that, I think, is that when we look at the region, we don't think it's just B4GALT2. There's actually another galactosyl transferase right beside it. And we think that two of these occur on a haplotype that's influencing platelet aggregation. But this haplotype is probably detectable in the Europeans, but dissolves in Africans, as well as has a completely different structure in Asians as well, too. So this is an ongoing area of study for us to kind of try to disentangle this signal in other populations. So I'm going to switch over now to a little bit of a technical sub-study, which is our work on CYP2D6. So I'm pretty sure everybody needs no introduction to CYP2D6. It's a highly polymorphic P450 gene, has over 100 variant star alleles known to it to date. One of the reasons why it's so challenging to interrogate those because of the neighboring pseudo gene that has about 97% homology between the two. So all the techniques that we use in our clinical laboratory is a long-range PCR that basically lifts the gene out of the genome and then subsequently interrogates it for all the known star alleles. But what we wanted to do is try to get an assay that can actually give us a much better full gene resolution sequence of the gene to help inform kind of more multi-ethnic novel allele discovery. So for that, or prior to mentioning that, I just want to say the frequencies of these alleles, again, is very variable across different worldwide populations. This is a very old review from 2003, but I think still can give us a nice idea of how variable the gene duplication, the loss of functional alleles are throughout different populations in the world. And I have to give a shout out to Andrea Gedick, of course, who is here, and her colleagues at PharmGKB, who recently published a much more granular view of these allele frequencies in the different worldwide populations. So our long-read sequencing approach uses PacBio sequencing. And the purpose behind this was to get more accurate and refined genotyping. We also wanted to have an assay that specifically sequences the duplicated allele, and that this would ultimately help us for novel allele characterization. So this is just an illustration of how it works. The papers just came out this year, so you can get more details there. But it basically pulls out the downstream copy of CYP2D6 and enables barcoding through nested PCR. What's important about it, though, I think, is that we can actually pull out the duplicated copy of 2D6 when it's identified. So we can specifically amplify that copy in addition to the downstream copy, put separate barcodes on them and pool them and sequence them. So that we did a lot of validation of the assay, but this is just the last table in the paper showing the utility of the assay. So these are known samples that have previously been reported that have unclear diplotypes based on the genotyping that was done. We have copy number plus a few other handful of samples that we have internally that are quite complicated. But when we subject them to our smart sequencing assay, we were able to resolve the diplotype of all of these samples. In addition, we found novel configurations as well as novel alleles, star 107 through star 109, have all been submitted to the P450 nomenclature page. So the reviewer asked us to actually try to figure out how many samples were revised in this study. And although the design wasn't really meant to do that, we did estimate that around 20% of samples actually had their 2D6 diplotype revised after smart sequencing. So the reason why I mentioned that is because this is something we're pushing hard on for multi-ethnic allele discovery. And so with Jun Yang, also back at St. Jude, his Guatemalan population that he's used for his ALL studies, we have MLPA data on for 2D6. Looking at copy number, he has all of the Illumina array data. We've actually finished sequencing them and found, this is just an example of one of the cases with the star 2, star 82 diplotype. Found quite a few rare alleles that previously have been underappreciated, like this one, star 82, for example, plus a few novel alleles that have strange configurations that Andrea is going to sequence for us with her allele-specific assays. A second ongoing Caribbean study as well, just getting going with Dr. Duconge from Puerto Rico, where he's got Guthrie cards from all around the island, looking at the different ethnogeography populations throughout the island of Puerto Rico that we're subjecting to smart sequencing as well too. So in the last few minutes, I was just going to go through our work on our favorite population, the Ashkenazi Jewish. Two of my favorite individuals are shown here. I'm quite proud of the fact that I get my haircut at the same barber on the Upper East Side as Mr. Allen here. But I have yet to see him. I've only seen his picture on the wall. He's rarely there. So the Ashkenazi Jewish population, it's the largest Jewish diaspora. About 80% of Jewish individuals are Ashkenazi. They originated in Central or Eastern Europe, and their population substructure is a composition of Middle Eastern, European, and African descent. They're very interesting if you look at the substructure. Of course, they overlap with the European CEU population when you have the Africans and the Asians in the mix. But if you remove those and just go strictly with the Jewish populations and the Europeans, they spread out. And so you see this nice climb between the Ashkenazi to the Europeans over here and all the different other subpopulations of Jewish populations up here based on their geography. But what I think is what's cool about this too, it's my friend Emer Kenny from Sinai put together, was that this climb across from European to Ashkenazi, you can actually determine the percentage of Ashkenazi these individuals are. If they have one grandparent, two grandparents, 50%, or three grandparents, or 100% Ashkenazi based on their PCA plots. So they're most well known for their high frequency of founder mutations for autosomal recessive diseases. But a few years ago, the Ashkenazi Genome Consortium published a whole genome sequencing study using 128 healthy Ashkenazi, and this was published in Nature Communications with Shai Karmi. Within this study was my friend Inga Peter from Mount Sinai, and so she came to me with the idea of doing a pharmacogenomic sub-study. we wanted to look at the novel variation in this population using a panel of PGX genes. So what we did was we went to our favorite resources, CPIC, PharmGKB, the PharmAdme lists, things like that, and came up with a fairly wide encompassing list of 377 genes, 23 megabases of the genome, about 0.7%, just to try to characterize some of the variation that might be unique to this population. So on the left is the distribution of all variants within these genes, and on the right is actually the over 16,000 novel variants that have not been cataloged in or identified in exact dbSNP, 1,000 genomes or any other available database, as well as their distribution in terms of their location. As you might guess, the majority of variation was rare. And this is consistent with what the 1,000 genomes taught us, is that the frequency of majority of variants was very, very low. And that usually suggested that it was more likely to be functional as well. And we can infer that by the graph on the right, which is the ratio of non-synonymous to synonymous. So it's more likely to be a non-synonymous variant at a lower frequency, which decreases with increasing frequency, which is also consistent with what we learned from the 1,000 Genomes Project. We wanted to do a quick snapshot of what might be clinically relevant. So what we did was we looked at the CPIC genes subset, around less than 10 actually. We removed HLA and 2D6 and things like that were difficult to interrogate by short read sequencing. And we excluded the known variants. So we took out STAR2, we took out STAR3, all those kind of big guns. And we want to see what else remained within these genes after excluding those. And this is the distribution here. We saw several hundred variants within these clinical genes. And again, the frequency of these variants was highly correlated with function. So this is using mutation assessor. So the darker the color here, the more likely these variants are to be functional. And then we took a random sampling from the 1,000 genomes of 128 individuals from the different European, South Asian, as well as the African population, and found a very similar pattern that within these clinical CPIC genes, there's a lot more variation after we remove the known star alleles, and that the more rare they are, the more likely they are to be functional. And this was also consistent within our AJ population with the novel variants as well, too. So with that, it's going to end on Ancestry and Pharmacogenetics implementation. I think we had a great discussion this morning, and I just wanted to kind of add to that, maybe if I can, by talking a little bit about Ancestry and some of these rare variants. So whenever I teach pharmacogenetics to our medical students, I always start with this picture and I ask them if they can tell me what it is. I don't know if anybody wants to shout out, but the rectangle in the middle over here should kind of give it away. So I work right here, I guess is the one way to give you the hint. So this is New York City, and this is an image taken from the census data. So every red dot represents 25 European individuals. Every blue dot is 25 African individuals. Every orange dot is Hispanic individuals, and every green dot is Asian individuals. And as a shout out to some of my colleagues also in this session today, here we have San Francisco, and here we have Philadelphia over here on the right. And I couldn't find Ferrara, Italy, unfortunately, or South Africa, so I apologize for that for the other two speakers. But we could certainly make some comments about health disparities and socio-demographic issues. But the reason why I point this out is to highlight, what is our local patient population that we're treating. So Mount Sinai, where we are, if we turn right down Madison Avenue, we are looking at the Upper East Side of Manhattan. If we turn left, we are at Spanish Harlem, which is a completely different demographic. As such, our patient population is extremely diverse. We have about 1/3 mixture of African Americans, Hispanic, and European individuals. So if we look at the variation in these different populations, and I think that it was very important to learn from this as we consider what variants are we implementing. So we looked at EXAC. Again, this is another study where we're looking at a large number of pharmacogenetic genes, kind of trying to characterize all the variation that we can see within these genes that have already been sequenced and the data is publicly available. And again, the rare variants are more likely to be functional than the more common variants. But this is what's important, I think, for this component of it, is if we look at the clinical genes, this is again going back to the CPIC genes here, how many variants per gene per individual are they in different populations? And you can see this nice pattern, it's kind of a U here. So it's African, East Asian, European is in the middle, Latino, and South Asian. So the lowest number of variants per gene per individual is in Europeans. So now we contrast this with the FDA-approved IVD clinical PGX kits that are available today, and the frequency of the variants that are in those kits is what's shown here. So obviously you see almost the reverse pattern. So we're actually clinically testing for the variants that are found within the European population, missing the majority of variants in other populations. So just highlighting the fact that it really behooves us to know what our patient population is that we're treating and what variants should be implemented when we consider pharmacogenetic implementation. So Mount Sinai has an implementation program as well. We've heard a lot about these kind of programs, preemptive programs already. These are the genes and the drugs. It's funded through the IPM, Institute for Personalized Medicine, as well as we had a eMERGE PGX component as well to it too in a parallel program. I live here at the Mount Sinai Genetic Testing Laboratory doing the genetic testing. IPM developed its IT infrastructure called ClipMerge that has the CPIC rules engine basically informing the rules that we pop up through the electronic health records. But I end on this because I wanted to just kind of comment on what to do with warfarin, because this is very relative or relevant to the ethnicity and ancestry discussion. And so we all, I think, know the story of warfarin and the challenges between the clinical trials that came from it, and the rationale being that the majority of African individuals in coag were actually missed. You know, the variants that they harbor are not those that were genotyped. They don't carry star 2, star 3, or the VCORC1 variant that majority of Caucasians actually do. Quite a few years ago, we identified star 8 as a variant that was at relatively high frequency in African Americans. Manoli Pereira has subsequently done some really elegant work on many more African variants found in this population. But how do we implement these variants into the warfarin story preemptively? So we just kind of came up with this logic that's tried to basically use the information from all the clinical trials that have been performed, as well as being smart about which variants we're testing for and what population this is in. And I realize this is hard for you to appreciate from the size of it, but basically it asks the question, is the data in the EHR? But then when it gets to here, this is basically asking what is the ethnicity of this patient? And if they're Caucasian, then they get kind of shunted over to this pathway where they're more likely to have pharmacogenetic guided dosing. If they're not Caucasian, then they get more likely shunted over to a clinical algorithm. So it's basically like in the coag trial, the age, the BMI, co-medications, and things like that. So we feel that that's a fairly good interpretation of the available data, but it is a complex issue. And because the reason why we can't actually have the other variants that are more common in African Americans and other ethnicities is that we're limited by what our clinical genotyping panel includes at this point. So I'll end there. My conclusions, I think, are going to be just fairly broad, is that we have the continued need for pharmacogenomics research among non-European and understudied ethnicities, and that long-read sequencing technologies are improving the ability to discover novel genetic variation. I think a crucial point here, too, is that the majority of pharmacogenomic variants are rare, and they're not currently characterized, and we're not really looking for them, or we're not testing for them clinically, and that these are actually more likely to be functional than the higher frequency pharmacogenetic variants. And then finally, clinical pharmacogenetic implementation should really consider ancestry-specific alleles based on your target population. And that finally, kind of on a negative, depressing note maybe, is that the race and ethnicity are increasingly difficult to define. So as I mentioned, we have our preemptive pharmacogenetic testing program with warfarin. We have 3 measures of race and ethnicity. One of them is when the patient is consented, they get to self-report what is their race and ethnicity. Another is their genetic race and ethnicity. They've all been genotyped in the biobank by multiple array platforms. But then a third is that what's in the EHR. And so when they go to their first clinical encounter at Mount Sinai, they either ask what is their race and ethnicity, or else somebody behind the counter actually checks off the box for them, essentially racially profiling what this individual might be. So you can imagine what those 3 measurements, what the Venn diagram of those 3 measurements look like. So it's very difficult to decide what is the most appropriate definition when including these variables into our pharmacogenetic dosing algorithms. So thank you very much for your time. I hope I'm not too late. These are the people I want to acknowledge from the department institutes, also in cardiology, and Alan Schulner from Regeneron. So thanks for your time.
So we have time for 1 or 2 questions.
That was a great talk. Thank you. My question is in regards to within a specific genotype, is there any evidence that there is a significant difference between different ethnicities for their response?
So within a given genotype.
Yeah, for choose your gene of interest, CYP2C19. I don't know that for a specific genotype.
Right. So is the magnitude different between different ethnicities?
Yeah, like is there any reason to believe that the response for a specific ethnicity may be significantly different than that for a different ethnicity for a given genotype, as opposed to the allele frequencies?
Right. So I would think yes. I don't have a strong example off the top of my head, but I believe yes, that there's, if it's a monogenic trait, then it's likely no, that the variant should be consistent between ethnicities. But if it's the more complex trait it is, the more likely there's going to be a variability between ethnicities given a specific genotype.
Thanks.
Yeah.
I'm kind of surprised to hear you say that. I don't know of any examples of that, but if you find one,
I know, I'm just making it up though.
Yeah, I think it's allele frequency more often, right? What's found, not the same genotype has a different phenotype in a different group. So I just wanted to ask about, you said it even at the end, it's very hard to define what race or category somebody is in, because if you have self-declaration, or if somebody assigns it by looking at somebody, or you do genetic testing. So what's your thought on just getting as many of these variants known as possible, but not testing someone based on their race, just including all alleles? Because I think occasionally we do have examples of people being in one category that might actually have an allele that you wouldn't think. I mean, I know that I myself have one of those.
Yes.
So I was just wondering, is that feasible versus?
So I think in the future it'll be easier because we just have full gene sequencing available to us. So regardless of if you're carrying an African ancestry allele or not, we'll be able to detect it. But for current, for the immediate term future, a lot of these programs are in kind of clinical research umbrellas. In the traditional clinical testing, the majority of that is limited by what's available as commercial testing panels. And most of those are very small in terms of the alleles that they cover. So I think for the next few years, there's going to be this in-between time where this is going to be really important. But once we get to a time when there's like clinically valid whole genome sequences available in your electronic health record, then that problem should go away, hopefully.
Right, I would think so. I just think it's dangerous to maybe type people and then genotype them based on that. Okay,
Yeah.
thanks.